Hey everybody, welcome to chapter 6. So in this chapter we're going to talk about viruses and then we're going to get into prions. We're going to talk about viruses, virions, and prions. So viruses were actually discovered, the first viruses were seen or discovered, I won't say seen, in 1892. Dmitry Ivanovsky was studying a disease in tobacco plants, which we now know is caused by the tobacco mosaic virus. He noted that the agent that was causing the disease was filterable, which means it could go through a filter. It was super tiny. So normally bacteria would be caught on the filter. So he thought maybe it was a really small bacterial cell, or maybe it was bacterial spores that were causing this disease. He wasn't sure. But one of his colleagues, Martinez Bergink, in 1899, noted that it was not bacteria at all. He stated maybe it's a poison or a toxin, and he coined the term virus. He called the substance a virus, Latin for the term poison. So that's where the term virus that we use today came from, because they said, well, it must be a poison of some sort. Well, we'll call it a virus. So viruses are super small. This is why Ivanovsky said maybe it was small bacteria, because when you look at our bacterial cells, bacteria in general are span around the 1 micrometer level, so super tiny, 1/1000th of a millimeter. But when we look at some of the largest viruses, they are smaller than some of the smallest bacteria. And so we can move over, and you can see there are some viruses that are so small, they are millionth, 1/1,000,000th or less the size of a millimeter. So they're super, super tiny. And so what is a virus? A virus is a non-living entity. That's why the term microbe is used to encompass all of the substances we study in microbiology. So viruses are non-living, but we can encompass the term microbe. We can encompass the virus in it. It's a non-living entity that can infect life forms, and viruses tend to have a limited host range, so they can only infect certain types of cells. Viruses contain only DNA or RNA. They never have both DNA and RNA. Their genetic material is surrounded by a protein capsid, and some have an envelope that surrounds them as well. The envelope actually is a target for a lot of our antimicrobials. If you target the envelope and get rid of the envelope, then the virus becomes non-functional. And so you all have heard of the coronavirus, I know, because of COVID-19. This is an enveloped virus. And so washing your hands using soap actually causes that envelope to break apart. And that makes the virus itself non-functional. That'd be a better word, right? Viruses are always obligate intracellular parasites. They cannot work outside of a cell. They don't have their own organelles. So they have to get into a cell, and then they take over the host machinery. And then when they do that, they can make new viruses, new particles known as virions that can then make brand new viruses. Okay. So the structure of the virus was actually found by Wendell Stanley in 1935. He is the one who figured out the tobacco mosaic virus was composed of RNA and protein. and he was able to see the structure using electron microscopy. He won a Nobel Prize in 1946 for his contributions to the field of virology. So we have two different types of viruses, naked. A naked virus has no phospholipid coating, that envelope, and then enveloped, which is like here, which contains a phospholipid layer. Enveloped viruses are easier to inactivate because once you get rid of the envelope, then the virus no longer functions. If you don't have an envelope, then you have to have other mechanisms to inactivate the virus. And this is just showing your naked virus and your enveloped virus again. I'm going to stop here, and we'll get into the different types of viruses in our next video. Okay? All right, bye.